Real pleasure to have Professor Christine Wells presenting to our MIG seminar series. So Christine is the founding director of the Center for Stem Cell Systems at the University of Melbourne. And you will see she has very good problems to solve in the computational and the modeling and the statistical field. So she's the architect of stemformatics.org, and it's a compendium of curated data sets in bulk and also in single cell for stem cells. And this compendium is for a lot of us who have been working with Christine, is a very great resource to do a lot of methods and data analysis projects. So without further ado, Christine, it's up to you. And just letting you know the audience that Christine welcomes interruptions. If you have any questions, you can just unmute yourself and ask. Otherwise, you can also put some questions in the chat and I can then translate the question to Christine.
Thanks so much for the introduction, Kim-Ann, and for the invitation to come and speak at MIG. I feel very, very synergistic with many of the MIG members, and so it feels a little bit like I'm speaking to family. And as Kim-Ann said, I really want questions as we move forward. So because I'm speaking to you about this idea that the sum is greater than the parts, I really did want to open with an acknowledgement slide, and in particular point out the people whose work I'm drawing on in this presentation. So in my group, I'm talking to you about work that's been developed by Paul Angel, who's a computational researcher in partnership with Nadia Rajab, who is a stem cell researcher. And a lot of the approaches to visualizing and exploring data from the Stemformatics team, which is led by Jani Choi. And a lot of the analysis approaches that we've taken have been developed very closely in partnership with Kim-Ann, and we're very grateful for her ongoing support of the research programs that we have. Yidi Deng, whose research I'll show snapshots of in this talk, was co-supervised by Kim-Ann and Jani. So I just wanted to lay out what I see the goals of computational cell biology are, which more and more I'm describing my group as sitting in. And that's really about building very detailed descriptions of the complex systems that make up human biology. When I'm talking about describing those systems, it's not just capturing the components, but also understanding meaningful relationships between those components. And that may be at the level of whole cells and tissues or down to very specific pathways and networks. We also have a goal to discover new classes of cells. We understand from recent Atlas projects that we don't yet have a firm idea about what all of the cell types in the human body are, and arguably where those cells transition between the same cell type in different cell states, or whether it's just a compendium of cell states. We want to be able to predict the behaviour of cells and systems ahead of time, and that's particularly true in the stem cell field when we want to control cell behaviour better. But also we want to use our descriptions to better classify our laboratory models of human biology. We want to benchmark new technologies and methodologies, as Kim-Ann alluded to. And I'm very interested in the idea that we can pull out the key components to design new cell types, new cell functions, and new cell states. So I come at this as a descriptive biologist, and my training was in the early days collating some of the atlases that make up our understanding of the human genome, gene content and gene output. And the focus of my research is in the transcriptomic domain. So, you know, early in the Atlas era, we set out to map the genome, and we really were convinced that we would be uncovering the the dictionary for human biology. And 20 years on from that, there's still an awful lot about the genome that we don't know. Side by side with that, we started mapping RNA content and RNA species in the FANTOM consortium. And as part of that cataloguing activity, we discovered new classes of genes. We discovered the complexity of the transcriptome in terms of isoforms. And there's still a large gap in our knowledge about how these new RNA classes and RNA isoforms impact on human biology. There have been huge efforts by other consortia to look at genotype-phenotype relationships, and increasingly we're understanding that we need to be looking not just at those relationships in the context of specific tissues, but also actually probably at specific stages in human development. And so being able to tackle some of these questions at the level of an individual cell is a hugely exciting new atlas that's coming out of the Human Cell Atlas Consortium. And there the promise is that our understanding between molecular characteristics and phenotypic characteristics can be synthesized at this level of an individual cell. I'm not going to go into all of the discoveries that FANTOM, the Functional Annotation of the Mammalian Genome Consortium, have led. Suffice to say that when you look at an individual set of experiments, you can make some anecdotal observations that are very hard to generalize without extensive experimental validation and verification of that anecdote. When you start to compile sufficient observations across sufficient diversity of cell types and tissues, those anecdotes become generalizable observations. And if the scale is sufficient and the statistical methods are robust enough, then those generalizable observations can become rules of molecular behavior. And one of the things I'm very proud about these genomic activities is that we do have much deeper understanding of the behavior of transcription and the controllers of transcription, the role of enhancers, promoters, non-coding RNAs in controlling not just the abundance of a transcript, but the stability of a transcript as well. And one of the contributions that I made as a PhD student in the first FANTOM project was that dynamic systems are really important to measure as part of these consortia activities. And this is from one of the publications from my PhD, where I was looking at mouse macrophages responding to bacterial product, lipopolysaccharide. And the figure just shows you that the genes that we knew about in 2003 were the genes which were highly abundantly expressed in a macrophage, but they didn't change very much upon stimulation. So the low-hanging fruit in 2003 were the transcripts which were very abundant. And the really interesting genes, the genes that said something about that dynamical response to bacteria, they were the genes that were essentially uncharacterized in 2003. Now, you would think that in the 20 years or so that have spanned that, we would have a much better grip on what the products of the genome do. And this is a back of the envelope calculation that I put together just a month or so ago by comparing pathway annotations in Reactome, which I think is probably the most authoritative source of pathways that we have at the moment, against the conservative protein coding set in UniProt. And you can see that just about half of the UniProt proteins from the human genome have no pathway annotation at all. And that a very large number of pathways in Reactome are actually represented by a very small number of genes. or proteins in this case. So 4% of the proteome hits a huge number of pathways. And this is a real problem for us because it drives this rediscovery trap. You know, we have gotten into this habit of running some kind of comparison between two samples or groups, getting a list of genes, and then essentially verifying that what we have is biologically meaningful by running it across a gene set enrichment analysis. And when we do that, we are inevitably finding the pathways that are already very well described. And we're not putting enough effort into understanding what this 48% of poorly characterized proteins are doing within a pathway context. I haven't even talked to you here about the non-coding aspect of the transcriptome, which is a much larger component in terms of the variety of species. It's also much harder to annotate a non-coding transcript to a protein because they lack the motifs that we usually do use to predict them in here. But nevertheless, it gives you a window on how little we actually know about the function of our own genome. And so when we're thinking about descriptive sciences and the role of atlases or catalogues in understanding our own biology, 20 years on from the human genome, we still lack a complete picture of the components. You know, we know that there are still gene classes to be discovered. We know that we have a very, very shallow understanding of transcriptional variation and isoform usage, even across tissues. We are very poor at predicting proteins which are below 30 amino acids. This was a very arbitrary open reading frame size set in the early days of genome annotation, and we really haven't improved on it. And of course, we now know that there are a number of transcripts which contain multiple open reading frames which can be translated, and the rules of that translation at the ribosome are poorly understood. So our biology is much more complex than we understand at just the catalogue level that we have today. And so further understanding how these are organised, particularly in a dynamic sense, so how complexes change as cells differentiate or respond to environmental cues, whether our pathway annotations are fit for purpose to understand this complexity, I think is another problem. And then of course there are levels of complexes that we can't easily predict computationally, such as RNA protein binding or RNA metabolite complexes, which I think are going to be very important in the feedback of a cell with its environment. And broadly, you might class these as post-transcriptional or post-translational modifications, but it's more than just understanding what's happening at an individual species, and rather understanding what's happening at the complex and in the system. And then, of course, we lack an understanding of the regulators. So we know that there is feedback between the level of messenger RNA and its translated product, but the rules don't hold universally, and we don't yet understand why. I've already alluded to the fact that there is metabolic modification of RNA. We really don't understand the extent of that modification or the context in terms of the mRNA stability or translation. And then we don't really understand the feedback of other physical properties of a cell, such as force or flux, shape or stiffness, with the transcriptional complexes that we're looking at. So, you know, those of you who are working in this kind of descriptive biology, there's lots to do. And that's great from a curiosity-driven perspective. But unfortunately, I think that the straightforward atlas approach, where we're just continuing to survey tissues, either at increasing molecular resolution by starting to bring in things like nanopore or some of the modification methods, or at increased cellular resolution through the single cell atlases, these surveys will not address the gaps that we're interested in. What we need to be doing is we need to be deep diving into specific experimental systems layering over as much multimodal information as we can within that same system. And so the trade-off, if we're wanting to build a lot of information very deeply on a system, is that we can't do that broadly across lots of systems simultaneously. It's just not feasible to do so, not within the current NHMRC and ARC funding scheme, nor, of course, within our capabilities to handle sufficient numbers of cells or tissues to do this multimodal activity on scale. And just to point out that we are actually moving our technologies for capturing information very, very rapidly, and yet our kind of management of the catalogue systems are not really keeping pace with these. Just 4 years ago, we started to see some early spatial resolution. Here I'm showing you some images of the mouse early embryo. So 2016 out of China, and Patrick Tan here in Australia, some very early spatial images of gene expression across the early epiblast. This is the very first time that the mammalian embryo starts to specify cell lineage, the cell types that make up the human body. And now on bioRxiv, we've just got this absolutely incredible complementary data set from John Marioni and Long Kai, which is, again, at this same very early embryo stage, partnering single cell transcriptomics with spatial transcriptomics, and also some collection of histone modifications as well. So starting to get this really exciting multimodal data. So if we're going to be relying on individual groups with great depth of understanding of the biology they're looking at to build these kind of miniature atlases, how do we then go forward and move from the anecdote to the generalizable observation to the rules of transcription that we were able to draw from the large-scale atlases? And that's where this approach of an aggregated atlas, I think, has to come in. And so just schematically, what I'm describing to you is that an aggregated atlas would draw from lots of different individual experiments, each of which is surveying a particular aspect of the biological system we're interested in to great depth, that in isolation, they are only showing one tiny part of that system, but en masse, we're able to build a much bigger picture of that system. And in particular, we're able to see patterns emerge that are reproducible across different experimental series, even though they've been generated from different laboratories, on different platforms, by different people, with different nuances. There are still some core biologies which emerge from those properties. And drawing on those core biologies, we can start to build some of the classifiers and benchmarking tools that we're interested in building. So stemformatics, the principle behind stemformatics is exactly this idea that if we curate sufficient depth of data, we can start to build insights into the systems that we're interested in. Stemformatics is more than 10 years in the making now. We went live to support Stem Cells Australia in 2011. We went live with 4 data sets and a lot of naive ideas. Since then, we have literally tens of thousands of samples which have been curated. We have launched a number of compilation atlases. We've collaborated globally. And most excitingly, I think we're starting to get to this idea of understanding how to bring together data that has been measured in different ways on different platforms to understand meaningful biology. The issue of curation is really critical. And of course, you're going to see lots of, maybe not lots, you're going to see other people who will take the approach of just grabbing in the whole of Geo or the whole of UniProt to start building these kind of aggregate series. But curating the individual experiments is incredibly important to understanding what you're starting with. And I'll tell you that in our curation process, we have a 3-step process. The first is we curate the experiment itself. Does it make sense? If it's setting out to address a particular problem, is it using the right cell benchmarks? Is it setting up an experiment that's not confounding, for example, batch and biology? Then we go through a QC process that assesses the quality of the data itself and asking whether or not the data is of good quality. Is it over-amplified? Is it sparse? Is it behaving unexpectedly? And then the third step is to actually look at the quality of the metadata. Can we extract the necessary information about the experiment from the metadata? Have the samples been assigned unambiguously, and so forth? When we go through this curation step, we fail 30% of the data that's in the public domain. 30% of published studies fail this curation process. And journal impact factor is no guarantee of data integrity. In fact, the very expensive Nature group have got a 70% pass rate in their studies, just as the impact factors of the discipline-specific journals do. And so I would urge you not to judge the quality of a data set or the quality of an experiment on the impact factor of the journal it's published in. It really has to be a process of curation of the experiment, the data, and the metadata to be sure that what you're getting is worth including. Some of the common mistakes that we see in individual experiments that would prevent them coming in easily to our atlas, very commonly is a lack of replication. This is really problematic because we accept that at times it's difficult to get a large number of replicates if the samples are very scarce or very expensive to obtain, but it's hard to work with no replication at all. And one of the motivations of building an aggregated approach is that perhaps an experiment which we dismiss in isolation because it's just n equals 1 might be of value in amongst the whole if we can find pseudo-replicates from other series. We commonly see experimental design that confound technical and biological groups, and there's just very little that we can do about rescuing this. Unfortunately, when people set up experimental design, they don't often think that mixing their biological groupings across the batches of measurement are really important. And this holds for single cell biology as much as it holds for microarray technology. We also find a lot of people who are trying to compare published and new data set up normalization strategies that predetermine the outcome. And so what I mean by that is that they might use such a strong batch correction approach that they pre-assign which groups belong to which biological class and force a change to the underlying data distribution or subtract sufficient information that you can almost predict the outcome before you've done the normalization. And likewise, if you want to show that a cell type is really similar to the cells you're interested in, using this very supervised normalization approach can actually inadvertently emphasize similarities. So we wanted to try and use some aggregation approaches that didn't set these prior expectations of the behaviours of the group. We wanted to expose the variance that might come from all of the technical as well as the biological contributions to the series. We also see very commonly that small numbers of markers are frequently used to assign cell types and cell classes. There is an opportunity to build really sophisticated machine learning classifiers, and this is one of the great things coming out of single cell biology, but people nonetheless tend to cherry pick those and use enrichment techniques to prove or not that the cells that they're looking at are the cells that they want. We've already spoken about the problem with gene set enrichment, so I won't labour that. And then surprisingly common is metadata mismanagement in the public domain, where data is either poorly described, where samples are given the wrong label, that happens surprisingly often, or data is simply missing. And accession hacking is a surprisingly frequent event where quite big name labs and papers will publish the control set of their data series, but not the experimental series that people are actually interested in and that they've described in the paper. So they need to have an accession number to get through the editorial barrier, but there's no editor checking that all of the samples are in that accession. So over the years, as we've run through this curation process, we started to tick up cell counts around different biologies. And you can see that there is a really strong emphasis here on some of the blood cell types. And that came in part because Stem Cells Australia had a blood theme, and we were supporting the curation of data in Stem Cells Australia, and also partly because my own laboratory is really interested in myeloid cell biology, and so we would try and scoop up as much of that data as possible. And so just a plug that you can have a look at some of the combined atlases in the STEMformatics site now. It's live. All of the data and the code is available for you to download and look at. And this is just another way of showing the kind of broad groupings of cell types that we have using the Atlas aggregation approach that I'm going to describe to you shortly, that Paul Angel developed for us. You can see that we've got a lot of data describing mesenchymal stromal cell biology, pluripotent stem cell biology, and of course here are our beautiful blood cells. We can then use this Atlas approach to hone in on subgroupings within one of these cell classes. And I'm going to describe for you how we build this and how we're using this now. This work is published, it's just come out in PLOS Computational Biology. All right, so the use case I'm going to show you for this aggregated approach, it is a description of two myeloid cell types, the dendritic cell and the macrophage, and I'll tell you why they're super interesting cells shortly. So I'll tell you why macrophages are super interesting right now. When I started my PhD, my supervisor said to me, "Christine, we are nothing but a big bag of water and macrophages. There is no disease that a macrophage does not have an involvement with." And honestly, I walked out of that meeting thinking, oh my God, I've started a PhD, I've committed 3 years of my life to a crazy man. And now I completely subscribe to that point of view. There is no human phenotype that I've come across that doesn't have a macrophage involvement. Macrophages are important for early organoid development, placental integrity, tissue homeostasis, you name it, pruning neural synapses, maintaining the gut microbiota, making new blood vessels. And so, of course, because of that, they have a role in infection and inflammation, in tumour surveillance, scarring, neovascularization, and lots of the chronic diseases that we have, lifestyle diseases of obesity and artery hardening, because macrophages form the foam cells that lead to arterial sclerosis. And yet, despite the kind of centrality of these cells and a lot of pathologies of human disease and a lot of the developmental features of human biology, we've got really bad models of human macrophages. Almost everything we know about macrophage biology comes from the mouse, or we've used circulating monocytes from PBMCs to differentiate in vitro the tissue resident cells that we're interested in doing. So one of the questions we wanted to ask with this aggregated atlas was, how good are these in vitro models of the real thing? So just reminding you that we're building this composite approach. And the way Paul conceptualized this was simply to ask, once we've got our curated data, which genes are really influenced by platform variations, so what's the batch component, and which genes are influenced by biology more. So we rescaled the data using a simple rank transform, and then ran a gene filtering approach based on this, the contribution of bacterial biology to the gene's behaviour across the entire data series. And doing this in a recursive way comes down to a small set of genes that have low platform confounding and high biological informativeness. Now, a lot of our batch correction methods assume that every gene is equally affected by the platform confounding effect. What I'm showing you here is just an array of genes ordered according to their sample source or platform variants. And you can see on the y-axis the proportion of variants explained either by the sample source, which is coloured in blue, or the platform, which is coloured in red, and the residuals are denoted in purple at the bottom of the graph. And so you can see that actually there are some genes where platform is overwhelmingly impacting on their behaviour when you pull data together, but there are lots of genes where platform is not the major contributor to their variants. So as we recursively filter the genes to remove this batch effect, here I'm showing you the blood atlas now, and the atlas is projected onto a principal component plot, and you can see that the platform is really driving grouping of the samples on this particular plot when we retain about half of the genes available to us. When we drop that down to just the 3,500 odd genes that have low batch contribution, you can see that we're mixing the platforms really nicely. And indeed, the groupings of the samples now have got a strong biological meaning, even though the annotation of that biology has been imposed after that filtering approach. And so we've made 2 atlases so far using this method, one describing the blood system and one looking in more detail at the myeloid arm. The myeloid arm, 3,500 genes, plotting just under 1,000 samples from about 40 independent data sets. And these have been measured across 14 different platforms, including microarray and population RNA-seq. We haven't yet included single cell into this atlas, but I'll show you some examples of projecting single cell data onto this atlas. And so I'm showing you now some great work that Jani Choi has done for us, where we can overlay the metadata that we have onto the expression data in this atlas. And in this way, we can start to build some condition matrices where we're understanding what the association between experimental observations or experimental conditions are with the behaviour of the genes that we're interested in. And you know, the statistical model that we are using here might be as simple as a regression analysis, or it might be using a simple Boolean model where you may want to bring a more sophisticated approach. But nevertheless, it all starts with the fact that we can build this condition table across a large number of experimental observations. And so one of the things that we found early on was that we could identify not just different cell types within this atlas, we could also see patterns associated with activation. So for example, you can see that one of the inflammatory genes that are associated with macrophage biology, interleukin-6, sits very much at the leading edge of one of these components. You can project your own data onto this, and this is really useful if you want to test out how good we are. You know, be a bit skeptical and try it out. But also if you want to start benchmarking new models of myeloid cell behaviour onto this atlas, you can see where they sit. You can also project any of the data that's hosted in Stemformatics onto the atlas as well. So I'm going to show you some examples of projecting single cell data onto the atlas and what we've learned about dendritic cell biology from that. And this is the work of a really talented honour student, Yidi Deng, who worked closely with Kim Ann and Jani Choi. So we've got two different single cell data sets here that would some of the earliest single cell data sets generated from peripheral blood dendritic cells. And interestingly, they did not agree with one another. One group described a set of dendritic cells which spanned the monocyte to dendritic cell phenotype, and the other group only described the DC2 category and were unable to see the monocyte category. And actually it came down to the way these cells had been prepared. So we were able to start projecting aggregates of these single cells onto the atlas and colouring them by their similarity to the different cell classes. And here I'm showing you the monocytes in that data series projecting right onto the atlas monocytes. And as we add the other aggregates together, you can see that we are indeed seeing that a class of dendritic cells spanning the monocyte to DC2 category. We're seeing monocytes that span the DC2 to DC1 category, and we're seeing a number of plasmacytoid DCs as well. Using an enrichment test, and here we've just used the capybara model from Sam Morris's lab. What we can see is that for the, I'm going to show you the PDC information because it's probably the easiest to see, that there are actually two groups of PDCs captured on our atlas with this single cell data. One group which is really well described in the literature already, and a second group which shares some overlap with the DC1s. And this subtype has been described before, but only seen in single cell data previously. And it's got pretty unique characteristics with regards to its responsiveness to some of the interferons. Right, so the next question we could ask with the this data set was, what are the tissue resident versus culture models of myeloid biology? And you might have seen on the backdrop some in vitro DCs which didn't look like any of the single cell data. And when we project in vitro data, we can see that our in vivo models of dendritic cell sit down here, and our cultured models of dendritic cells sit out to the side here, and our projected models also sit quite close to cultured monocytes. So what we're seeing is that the culture models of dendritic cell biology do not approximate the in vivo models at all. That's just summarising that for you, and I'm happy to describe it in more details for you later. So the second type of dendritic of myeloid biology that we're really interested in is the macrophage. And here I'm showing you some macrophages that Nadia Rajab, very talented student in my lab, has made from iPS cells. And they're doing all the things you expect a macrophage to do. They wander around the dish, they're gobbling up the debris in the dish. They express the markers over macrophage, and they respond to macrophage cues. But when we look at the behaviour of monocytes and macrophages, either differentiated in vitro from a monocyte or from a macrophage, we start to see, again, this culture phenotype coming through. And in fact, on this very busy slide, what I'm showing you is just a comparison of one of those groups. So these are monocytes which have either been profiled directly from blood or have been profiled after spending some time in a culture dish. This period of time may be as short as 2 hours or as long as 3 days. But what you can see is a very profound shift in their behaviour on our atlas. And you can see that we've got lots of individual measurements of these phenomena by seeing the number of samples replicating these behaviours for different genes. And when we look at the classes of genes that separate these two groups, we can see that they are the classes of genes associated with monocytes rolling along blood vessels and starting to move into tissue, where they are losing some of the selectins associated with that rolling and upregulating some of the receptors which are part of the chemotaxis into tissue process. So culturing a monocyte actually starts to mimic this rolling and diabetes behaviour. When we look at the stem cell derived macrophages, so the macrophages like the ones Nadia made from an iPS cell, we can see that they sit up here. So there's a clear axis on our atlas which distinguishes in vivo and in vitro derived cells. Again, using this conditioned matrix approach, we could build some regression models to show that the genes that capture this axis really fall into some very surprising categories. So the in vitro derived macrophages have got this ectopic expression of collagens and extracellular matrix proteins. Nadia went on to demonstrate that not only are they expressing the transcripts, they're also expressing the protein, and this is not something we're seeing in a cultured monocyte. And then very surprisingly, she's gone on to show that if you change the conditions of the progenitor cells, you can impact on the expression of this ectopic network, but only if you do so in the right developmental window. So if you try and impose that same condition early or late, you don't change the behaviour of this ectopic network, but if you hit the right developmental window, you can turn it off. So, you know, differentiating cells, stem cells into particular cell types like a macrophage is quite a complex process in the laboratory, where we're trying to mimic the developmental exposures of early lineage patterning genes, so some of the Wnt and BMP pathways, and then specifying the mature cell type by adding in these growth factors. And we're doing it over a very contracted time frame. You know, the developmental process for a human would be 9 months. Here, we're doing it over a very protracted time frame. And so the length of time of exposure of these factors and the concentration of these factors are important. And what Nadia has shown is that if you hit these cells at the right window in this series, we're not only able to turn on the macrophage genes, but we're also silencing the residual genes associated with that mesodermal patterning that are not being silenced otherwise in any of the public models of iPS-derived macrophages at the moment. And this actually really excites me because it tells us something new about how some of these phenotypes in cells are being imposed on the cell during development. And so we often talk about genes sitting within networks and that these networks coordinating a function. But what we don't often consider is how they are prescribed through development and whether there are windows where these networks can be manipulated, for example, to produce cells that have hybrid or alternate functions. We've accidentally made a cell here, an iPS macrophage, that has both some features of the macrophage and some features of its parental mesoderm. And we've also shown that we can control the development of that network. And so it's feasible then that there are other networks, and indeed we've got a lot of evidence that there are other networks that are imposed sequentially on the developing cell as it moves through that developmental hierarchy that might be co-opted into other cell types or provide an ectopic pathway for further functional exploration. So the lessons I hope you will take away from this combined Atlas talk is, first of all, that comparisons that don't adequately contextualise the biology aren't useful. And so we have to move on from just doing broad, shallow surveys of the tissues that we're interested in and start actually bringing truly dynamical and multimodal systems together. So we need better ways of aggregating data to understand what's happening across our systems. Be very wary of the supervised normalization approaches, which are standard approaches when bringing data together. They will bias the outcome. I hope I've shown you that by integrating a lot of this different data together, we can start revealing new properties of a system, things which may be anecdotally observed or actually not a strong enough signal in isolation are now able to be discovered and validated en masse. I also think that the older studies that we've collated have this very rich and useful metadata that can be used to annotate newer data types. And so we're using our myeloid and blood atlases as a reference to annotate immune cells that may be present in single cell data sets. So I'm not going to spend any time speculating on where I see future challenges for computational cell biology are. I just want to finish again by thanking a fabulous team of people, and in particular, the people whose work I spoke about today, Paul, Nadia, Jani, Yidi, and of course, the wonderful Kim Ann. Thank you very much.
Thank you, Christine, for a very great talk. Do we have any questions for Christine? Feel free to unmute yourself.
Hey, Christine, I had a question about the, I was really surprised by the impact that different platform types seem to have on the gene expression data. It seems absolutely mind-blowing to me. Yeah. So I was wondering, when you said some genes seem to be more impacted than others, sorry, of course I get a phone call right then. Is it the ones, is it like the really highly expressed genes are much more robust across all the different platforms? So it's not a gene expression level thing. Okay, all right.
No, I mean, that's a smart question, Andrew, and it's one of the first questions I asked Paul, because I was convinced that would be the case.
Yeah.
And it's not, surprisingly. I think it just comes down to how we measure gene information. So some of it comes down to the fact that you've got a continuous measurement on a microarray, and it's the quality of the hybridization that counts the strength of the signal, whereas with RNA-seq, you know, it's library depth and perhaps also library construction that are making the difference. So you're starting with data structures which are fundamentally different and are actually being surveyed on different scales as well.
It's hugely concerning seeing that picture. It's disturbed me a lot.
Well, it's surprising, Andrew, because a lot of biologists think that the biology will be the dominant activity. And, you know, if this is a biology that's reproducible, it doesn't matter how you measure it, right? Yeah. And I think every data scientist in the room is going to be screaming at me, but batch, because it is really hard. It's a really big confounder.
Yeah. Thank you.
So I have a follow-up question on this. So does that, and it's, I don't have the answer, I've thought about it for quite some time, but do you think it's because some of the genes that drive the biology have less batch effect in a way? You know, what is happening here?
So I think the strength of correlation of some genes with the biological class so outweighs the batch. I think that's certainly true, but it's not universally true, Kim Ann. What we know, actually from work that I didn't present today, but from work that another of Jani and my students has been looking at, Jinli, that you can bring cross-species atlases together if you use curated sets of lineage markers quite nicely, but you don't automatically recover those lineage markers when you use this unsupervised approach, because some of those lineage markers, maybe it's representation in the population you're looking at as well. But it did surprise me that there was not a, if you've got an impact of batch on an experiment, that it's not uniform across every measurement in that experiment. That really surprised me.
Yeah. Other questions for Christine?
I've got a question for Christine. Christine, now I'm on my rowing machine, so now I can see the whole thing. But anyway, so the question was really, how agile are these atlases? Because there was this nice set of 2 papers by Miki Epesuya and James Briscoe a couple of months ago now in Science, where they looked at the effect of protein degradation in driving cell fate decision-making. Is that something, certainly these atlases can help them analyze their data, but how agile is an atlas like yours to incorporate these new types of data in the analysis or in the structure of your atlas?
It's a really great question, Michael. It's something I've been thinking a lot about because, understanding protein turnover and how that might be reflected in the transcriptome, that's a big question in itself, right? Actually, the answer to that, I would point to Jess Ma, who is a statistician at the University of Queensland, who's been doing some really nice modelling of expression variance across the atlas measurements as an indicator of transcript stability and perhaps some of the regulatory constraints on those transcripts. I'm not sure that she's done a direct comparison between that expression variability and protein turnover, but it would be an interesting correlate. But I think essentially if you can get your data into a matrix where there are easy points of reference to bring it together, maybe a gene identifier or whatever, that it could be included. I guess the question about how sparse the matrix then becomes will be a really interesting one.
I think in their case it would be super sparse. There would be hardly any, so they're very, very targeted analysis, I think.
Perhaps, but if you're just interested in a pathway or you're interested in a small phenotype, that might be okay.
Yeah.
Other questions? Could you comment a little bit on using bulk as reference? Because I think it's quite, it's shifting the paradigm a little bit now that we're all entering single cell.
Well, look, I think it's no secret when we have single cell data that it can be very hard to identify all of the groups that we cluster in that single cell data set. And we frequently rely on the kind of gene set enrichment tools to assign cell types based on the expression of a small number of markers. And for some really well-described cell types, that's okay. But it does fall a little bit into that rediscovery trap. I think if you can have a reference data set that is bulk, so it won't capture all of the heterogeneity within a particular cell class, but it will capture the common reference points or signatures that define that class, then you can at least get a position of what type of cell it is before moving on and dissecting out cell state at that single cell level. Too frequently, I'm seeing cells which are misassigned or which are not assigned at all because the reference that you're using or your knowledge of those cell markers is just too anecdotal. But actually, I'm interested in hearing from other people in the audience who are using single cell data about their experience. using references like this versus sets of signature genes, for example.
Do you think that it means that the results that Nadia got, she couldn't have obtained if she only analyzed the single cell data?
Well, the difficulty, if she just looked at the single cell data, is that they would have looked like macrophages, but we wouldn't have known how different they were from the reference. And so, again, if your goal is to say, here are the cell types I'm expecting, here are the cell types I've obtained, you're really just enumerating the cell classes, but this is going a little bit further and actually assigning some kind of relationship measure or distance between those cell classes and the cells you've actually obtained.
Okay. Any other questions for Christine?
You can ask Johnny or Kim Ann too.
Okay, so I think if we don't have any other questions from the floor, let me thank Christine again for her excellent presentation. It was really good to have you. And that's it. We see you all very soon for the next seminar series. Thank you, Christine.
Such a pleasure. Thank you all.
Thank you. That's great.
Thanks, Christine.